Hello, I am Samantha Lewis, a Master's student of Animal Welfare Science, Ethics and Law at the University of Winchester. I will be discussing the welfare issues related to captive sugar gliders and the social structures we implement for them whilst maintaining and improving their welfare. Sugar gliders get their name from their diet of sugary eucalyptus sap, nectar and pollen alongside insects and their honeydew, combined with their patagium allowing them to glide. They are small marsupials who give birth to complete development within the mother's pouch. Longevity was believed to be 7 years in 1973 by Smith, increasing by 2010 to 12 to 15 years. However, this initial figure was early on with limited research and experience. The number may have increased due to better husbandry and diet, or may be attributed to more experience and a more accurate average. They are classified as exotic pets because they differ from normal companion animals and have not gone through domestication. Domestication requires several generations and physiological as well as biological changes. Misinformation suggests that several generations into their captive breeding means they are domesticated by Cooper, but this is inaccurate. Sugar gliders are colony animals with a variety of group structures, but generally consisting of a male and multiple females with their young. The colony requirement is important to sugar gliders and their choices, and timeframes need to be implemented within captive populations to ensure good welfare, which is often not considered in these companion animals. Looking at the suggested checklist for species suitability to captivity, we can see in 1.5 that this social interaction and natural behavior need to be provided for, alongside the 5 domains model, whereby domain 5, mental state, specifically interactions with other animals, must be considered to ensure good animal welfare. Sugar gliders are endemic to Australia and New Guinea forests, as seen on this map, with thriving populations introduced into Tasmania. There are multiple subspecies suggested with conflicting support, and regional overlap allows for potential crossbreeding. The map shows the regions for the 7 subspecies. Also, through genetic analysis, it has been noted that there is strong distinction between Indonesian and Australian populations. Although native to Australia, most states do not allow them to be kept as pets or permit exportation. Recent research has shown that sugar gliders may in fact be made up of 3 species: the sugar glider, Crefts glider, and Savannah glider. These discrepancies show a clear need for further research within wild populations and have the potential to affect the IUCN Red List status. Currently classified as least concern, smaller distinct populations may be under greater need for protection. This in turn could lead to tighter regulations on captive populations. There would need to be greater onus on the breeder's responsibility to discern their glider's genetic background and breed pure lines to ensure a captive population safety net and justify their captive presence. These subspecies, species, and regional distinctions are important with regards to captive populations, as apart from the limited study in the US, it is unknown what species or subspecies may have been crossbred within captive populations. Although subtle and not observed by humans, there could be communicational differences confusing individuals as a different dialect almost, causing further issues with communication and forced colony members struggling to maintain stability. This could be further exacerbated by offspring not knowing where they fit in between subspecies, if they receive conflicting behavioral predispositions. Communication is especially important in gregarious or group-living species. Some sugar gliders, including captive individuals, have differences in head size and shape, as seen here, which may further suggest communicational issues between different lines when combined with differences in auditory bullae, which is part of the inner ear. Key anatomical differences such as these may create miscommunication where subspecies are mixed, leading to aggression or unstable colony structures. Sugar gliders' auditory communication consists of a variety of sounds, where there is believed to be good understanding of their meaning, as shown in this table. Olfactory sensors are also important to the sugar glider as a chemical signal being sent and received between individuals and allowing behavioral or physiological responses, with the sender benefiting from doing so. They can differentiate between species and individuals on scent alone, sharing information regarding sex, reproductive status, and mood. This marking is generally carried out by dominant males, who mark females within their colony and objects to distinguish territory. It is suggested by Smith that scenting is a non-aggressive method of establishing and maintaining hierarchy, allowing for less costly establishment for all males by reducing fighting. These scent glands are located on the forehead, throat, and paracloacal glands, as demonstrated in this image. Scenting can be exacerbated in captivity. Having a strong smell, especially in tired males, many human guardians frequently clean the entire cage to remove the smell, resulting in the animals reapplying their scent to the enclosure and a vicious cycle ensues. It is highly likely that this situation is stressful. The animal wakes to find their territory is no longer designated their own. This welfare issue has been alleviated within the companion community by cleaning separate parts of the cage on alternating days, or using vivariums to contain the scent instead of open wire cages. Unfortunately, these solutions are implemented with the human in mind and not the animal's welfare. This flowchart gives a simple overview of early developmental stages in wild sugar gliders with free choice. a vital point that if they choose to leave the colony, they do not do so until around a year old. Twins occurred 81% of the time, and the remaining 19% being single joeys with anecdotal suggestions of triplets in captivity. These twins are ideal for rehoming together as they ensure familiarity, but this doesn't allow for choice. The study in wild populations showed 50% of young dispersed and were recruited into other colonies, but this occurred when the young were at least a year old and only females were seen to remain within the natal colony. This clearly demonstrates that given the choice, the gliders choose to stay in their colonies longer than they are given the opportunity to in captive environments. Conversely, females in captive breeding colonies will regularly breed year-round. In the wild, they have a breeding season spanning July to September, producing one lot of young. The presence of these young may give rise to the prevention of more offspring, or it may be due to the consistent temperatures and food allowing for this breeding practice in captivity, but it does raise welfare questions regarding the demands on the female and the stress from colony separation. Wild populations of sugar gliders vary in their structure but are considered polygamous, whereby one male will breed with multiple females. This table gives some idea of the variety of wild colony adult structures when free choice is available, with an average of 7 adults and 1 to 3 young. 75% of the observed animals lived in colonies, with the remaining 25% only being temporarily alone. Sadler and Ward noted male codominance to be between closely related males to ensure genetic payoff for both males. This cooperative behavior is evolved for reciprocity, mutualism, or as in this case, kin selection. This ensures that the benefits of maintaining the colony together outweigh the costs, whereby the close relatedness of the males ensures the genes are still passed on if they help ensure their relative reproduces. This includes the observation of a long-term father-son coalition with 2 unrelated females. High social status within hierarchies is important within colonies, as dominant males will father at least 75% of offspring. Even within wild colonies, individuals are rarely observed interacting with one another outside of the nest. This could be limited due to human observation being inconsistent, or the animals focusing on foraging and hunting. It may also be behavior born out of necessity, creating colonies to maintain warmth in the nest. However, they do interact with one another in captivity, with a singly kept glider developing concerning behaviors linked with poor welfare. Predominance and hierarchies have also been seen in captive colonies, but this comes back to issues regarding choice and space constraints. Confrontations to establish and maintain hierarchies can be diffused by the ability to escape, which cages do not allow for. Several captive studies have been carried out on male sugar gliders, but these have included small sample sizes of 9, of which one male self-mutilated when they were housed singly. In such highly social animals, this was against their welfare and could have affected the results, and was seen in Bradley and Stoder's 1997 experiment regarding testosterone and dominance links. This experiment further confounded the results by only allowing short recovery periods between surgery and stressful situations. This involved them being placed into a pre-established colony, thus creating an accumulation of stress load and causing potential distress, whereby the introduced male glider could no longer cope. These captive environments and experiments do not allow for animals to retreat from aggressors or find alternative food sources, and as the results demonstrated, can cause weight loss. Anecdotal accounts suggest pairs are common in companion animal homes, consisting of male-female pairs, with males entire or castrated, female pairs, or castrated male pairs. Studies have shown that males create a hierarchy, with subordinate males having decreased testosterone but increased cortisol, a hormone linked with stress. The study entailed forming colonies consisting of 6 unfamiliar wild-caught males. This was unnatural given the lack of all-male colonies in wild populations. However, as aforementioned, wild males have opportunity to escape each other. Stable colonies and female hierarchies are rarely studied, and even then consist of small sample sizes and therefore are not representative of colonies. This study looked at cortisol but excluded subordinates and their welfare. Studies need to be conducted on cortisol levels with subordinate males, preferably fecal samples, as these are less invasive than blood samples and therefore do not confound the results during sampling methods. The sugar glider is a highly social animal and has been known to bond well with the human guardians, adding to their appeal as companion animals. However, emotive language saying that minimal physical contact with its owner suffices for their needs, or that they can be maintained singly if they receive enough of the right sort of attention, gives damaging misinformation for sugar gliders' welfare. Furthermore, marking of guardians can be attributed to marking of the animal's belongings and not a colony member. Human guardians make poor substitutes for conspecifics, and the animals are under no disillusionment that their guardians are of the same species. Humans cannot provide the vital playing and feeding behaviors that are so intrinsic to sugar glider welfare, which fellow conspecifics give. They need appropriate companions and protection from isolation and unstable colony structures. Behavioral and health issues resulting from isolation include biting, vocalization, and self-mutilation. There are anecdotal methods of introducing sugar gliders to form new colonies. Unfortunately, these are often ignored, risking injuries and the health of all animals involved. The stress of moving gliders and placing them into a new environment can lower their immunity, thus the presence of normally controlled pathogens can get a foothold. A healthy animal can quickly become ill and put other gliders at risk by shedding these pathogens. This is true even with trusted breeders, and is why quarantining new animals is essential. After quarantining, it is further recommended to do slow instructions, as an established colony can do substantial damage to an unfamiliar glider. During this period, new animals are supposed to have their own appropriate enclosure, but this is often not enforced. Many guardians have got their new gliders home and put them straight into established colonies' cages, a clear violation on their welfare. It is important that the onus of this lies with the breeders. They must see evidence of enclosures and, when selling single gliders, evidence of current gliders. They must also offer support and guidance regarding instructions and quarantine, remembering that this is not about having disease in their colonies, but allowing the animals time to recover from their stress loads. Then there comes further issues when one of a pair inevitably dies and leaves the remaining animal alone, whilst the cause of death is ascertained, during the procurement of a new companion and the quarantine introduction period. Assuming there are no complications in these unfamiliar animals bonding, this period can span months and can create health and behavioural issues in both animals. It should therefore be encouraged that colonies consist of no less than 3 animals, instead of 2, allowing for time to increase the colony size again, with minimal stress on the animals. Breeding colonies consisting of a male-female pair and castrated male should be encouraged, allowing a natural but safe hierarchy to form and giving the parents an additional helper in raising offspring. There have been numerous studies carried out on male hierarchies in sugar glider colonies within wild and captive populations, but limited application with females or across the entire colony. These issues within captive sugar gliders raise multiple welfare issues, which as an emerging field has very limited jurisdiction in these studies currently. From the initial choices of colony members, which animals remain or leave, and at what age they do so, the colony structure of male to female ratios, and the dominant or subordinate status. The individual animal's responses, and not just that of the species, is important. Breeders often remove young gliders at 10 to 12 weeks old, long before gliders would opt to leave. Removing individuals does not allow females the choice to join the colony, or those who are kept the opportunity to leave. This is done due to space constraints, allowing guardians to have young gliders, and prevent father-daughter matings and father-son fighting. In the wild, inbreeding is not an issue as dominant males lead colonies for 2 years, thus reducing inbreeding opportunities. This 2-year dominance needs to be studied further, as this could give a natural solution when combined with a polygynous nature, whereby rotating males between breeding colonies to prevent inbreeding. Although not allowing for choice, it does mean young gliders can remain in their natal colony longer, alongside castrating males that are not intended for breeding. The industry standard is 70% of exotic pets die at wholesalers, with no respect for an animal's intrinsic value. This number is considerably greater when including mortality from capture stages, with considerable welfare burdens, especially with regards to social disruptions. Fresh genetics does not justify such a high cost, especially where carefully recorded lineage would suffice. Well-managed captive breeding with legal importation has thus far given the captive population greater genetic diversity than the thriving introduced Tasmanian populations, so it is feasible to maintain these populations forthwith without further captures. It is estimated sugar gliders were first brought into the US pet trade in the 1990s. Although now illegal to export and own sugar gliders in Australia, there has been speculation that many captive sugar gliders originate from either illegal capture from Australia, sold under the guise of legal trade from Indonesia, or exports predating Australia's laws. However, following DNA analysis, captive animals in the US originated from legal regions. If this genetic analysis is an accurate representation, they came from a small population, and therefore there shouldn't be an issue of communication. But this in turn poses welfare concerns regarding inbreeding and healthy genetics. This has created arguments for wild-caught individuals to give genetic diversity within captive breeding, which Campbell et al. Study supported. It would be important to analyse DNA of any imports to ensure they are from the described regions and further continue pure subspecies lines, enforcing this as part of importation regulations and committal of new animals to an extensive register. This study demonstrates that analysis of captive gliders can be implemented to ensure pure subspecies lines are maintained. These results do need to be taken cautiously though, as it was an opt-in study. Those breeders knowingly using illegally obtained animals are unlikely to participate, or select only their legal animals. Also, just because the animal may be from a legal region does not mean it was legally caught. There are export quotas permitted in West Papua, Indonesia. However, there is no requirement for documentation to state where they were caught. Furthermore, it also needs to be considered the effect it has on wild colonies when individuals are removed. What can we do? More studies focused on improving welfare with larger representative samples for all colony members. More pressure on breeders to acknowledge their responsibilities, including vetting prospective guardians to ensure gliders are not being kept singly and that correct quarantine and introduction procedures are followed. Also supplying care sheets and continued support. Changing the accepted minimum sugar glider colony size from 2 to 3 animals. Implementation of licensing focused on knowledge and education to discourage illegal trade. Rotating male between breeding colonies to allow young joeys to stay longer with a colony and castrate young males if not being used for breeding. Remove or reduce wild capture, alongside stricter regulations including the need for genetic analysis to keep subspecies pure. Create a worldwide lineage system. Have a standardized husbandry guideline which grows and evolves with experience to help minimize misinformation, to acknowledge what is best for the sugar glider and not just the guardian. It is clear that misinformation and non-representative studies are rife, largely focusing on males. The multitude of subspecies, potentially species and regions may lead to complications in communication and crossbreeding in captivity. There is also a need to enforce the presence of conspecifics and allow colony choice, whilst accepting the variety of natural colony structures, which can be achieved by reputable breeders. There have been several suggestions made to ensure improved welfare, most notably the minimum colony size increased to 3 and moving away from wild-caught animals by improved lineage records, whilst ensuring the needs of the animal are met before that of the human guardian. Thank you for listening. All photos used were my own.